Chemokines. Chemokines, chemotactic cytokines, are a specialized family of small signaling proteins, 8 to 12 kilodalton, that regulate the migration, chemotaxis, and positioning of immune cells. They are a subset of cytokines but specifically control cell trafficking. They bind to G protein-coupled receptors, GPCRs, on leukocytes. Their main role is to direct immune cells to sites of inflammation, infection, tissue injury, or lymphoid tissue for immune surveillance. Structure of chemokines. Size: 7125 amino acids, 8 to 12 kDa. Disulfide bonds: 4 conserved cysteine residues form disulfide bridges, which stabilize the structure. Folding: mostly beta sheet with 1 alpha helix. Receptors: bind to 7 transmembrane domain GPCRs. Key feature: arrangement of the first 2 cysteines determines classification. Properties of chemokines. Small proteins, 8, 12 kDa. Basic in nature, bind to negatively charged glycosaminoglycans on cell surfaces / extracellular matrix. Secreted in gradient form, guide directional migration, chemotaxis. Bind GPCRs, activate signaling pathways, calcium flux, actin polymerization, cell migration. Redundancy: many chemokines can bind multiple receptors, and one receptor can bind multiple chemokines. Functions of chemokines. Leukocyte trafficking: recruit immune cells to sites of infection / inflammation. Example: IL-8, CXCL8, attracts neutrophils. Lymphoid organ development: help in T and B cell homing to lymph nodes. Example: CCL19, CCL21 attract T cells and dendritic cells to lymph nodes. Wound healing: recruit fibroblasts, endothelial cells for tissue repair. Angiogenesis: some chemokines promote or inhibit new blood vessel formation. Hematopoiesis: influence bone marrow stem cell mobilization. Cancer biology: tumors exploit chemokines for angiogenesis and metastasis. Classification of chemokines. Chemokines are classified based on the position of the first 2 cysteine residues. CXC chemokines, alpha chemokines. CXC rightward arrow 1 amino acid between first 2 cysteines. Many attract neutrophils, lymphocytes. Example: CXCL8, IL-8 recruit neutrophils. CXCL10, attracts activated T cells. CC chemokines, beta chemokines. CC rightward arrow 2 adjacent cysteines. Mainly attract monocytes, T cells, NK cells, eosinophils, basophils. Example: CCL2, MCP1 recruit monocytes. CCL5, RANTES recruits T cells, eosinophils, basophils. C chemokines, gamma chemokines. Only 2 cysteines, first and third conserved cysteines missing. Rare group. Example: XCL1, lymphotactin attracts T cells, NK cells. CX3C chemokines, delta chemokines. CXXXC, 3 amino acids between first 2 cysteines. Only one known, CX3CL1, Fractalkine. Exists in both membrane-bound form, cell adhesion, and soluble form, chemotaxis. Attracts monocytes, NK cells, T cells. Examples of chemokines and their roles. CXCL8, IL-8 neutrophil chemotaxis, acute inflammation. CCL2, MCP1 monocyte recruitment to inflamed tissues. CCL5, RANTES attracts T cells, eosinophils, basophils, allergic response. CXCL12, SDF1 stem cell homing to bone marrow. CX3CL1, Fractalkine adhesion and migration of T cells, monocytes, NK cells. Importance of chemokines. Immune defense, direct immune cells to infection/injury sites. Homeostasis, maintain immune surveillance and lymphoid organ architecture. Inflammation, key mediators of acute and chronic inflammation. Wound healing, recruit repair cells for tissue regeneration. Disease relevance. Autoimmunity, aberrant chemokine expression, rheumatoid arthritis, multiple sclerosis. Cancer, tumor growth and metastasis via chemokine signaling. HIV, uses CCR5, CXCR4, chemokine receptors, as co-receptors for entry. Therapeutic targets, blocking chemokine receptors, example CCR5 antagonists for HIV treatment. In summary, chemokines are small, structurally conserved proteins that act as the GPS system of the immune system, guiding leukocytes to precise locations. They are classified into CXC, CCC, and CX3C families, and play crucial roles in immunity, inflammation, wound healing, angiogenesis, and disease pathogenesis. Thanks for watching.